Okay, just a quick podcast. A really great question came up by one of your fellow students, and I thought it was just worth clarifying. So we're going to be talking about MHC Class 2 presentation. And the question was kind of how does everything get into the same compartment? Okay, so and I kind of just gave a real brief overview in class because maybe it was covered in cell bio, but just to kind of get everybody on the same page. There's a couple different ways that exogenous, so this is MHC Class 2, so there's a couple different ways that exogenous material can come into the cell. Okay, and so it can come in by a process called endocytosis. And when it comes in by endocytosis, like a receptor gets endocytosed, okay, it's contained in a compartment called the endosome. So we have endocytosis. We also have the ingestion of big stuff like whole bacteria. So that comes in to the cell by phagocytosis. Okay, so in general, so here's our bacteria. This is the way to bring material into the cell. Just make sure that's all on screen. So the ultimate trajectory of these, this is called the phagosome. The ultimate trajectory of the endosome and the phagosome is basically to fuse, to acidify and fuse with a compartment called the lysosome. At the same time that this material is coming right from the outside and coming in, right, we have our ER. Here's our ER, and this box will be our Golgi. We have secretory vesicles that are leaving this compartment, and they contain proteins. Okay, these are secretory vesicles, and they too can be tagged eventually for the lysosome. Okay, so what happens is that these compartments meet up. So maybe this one is carrying, in this case for MHC Class 2 presentation, this one is initially carrying the alpha and beta chain of MHC Class 2 with the invariant peptide bound in. Okay, so there's our invariant peptide kind of plugging up that so that other peptides can't get in. And so this material, as it acidifies, it gets digested, right? And then as this compartment kind of acidifies into the lysosome, right, this becomes, the invariant chain is also degraded. So here's our alpha and beta chain, and then we're left with clip in here, okay? And then at the same time, this is fusing now with this chewed up, either in this case, some antigen that was picked up by a receptor or some of this pathogen that got chewed up into bits by the lysosome. These all kind of fuse together. So now we have peptide in here. And this clip is exchanged for peptide by HLA-DM. And of course, HLA-DO antagonizes this. Are we on screen? Good. Okay, so now we have here, headed back to the membrane now, our alpha and beta chain with the peptide bound in. I guess the peptide should be this color, and this cycles back to the membrane. So we have things coming into the cell from the outside, but also the alpha and beta chain are coming in from secretory vesicles from the Golgi. And these are all kind of meeting up in the lysosome and then traveling back to the membrane. And so I thought this was a really great question about how does everything come together, and this is the full picture. So essentially things come in from the outside and meets up with the secretory vesicles containing class 1 and class 2, and those came out of the Golgi. Those meet up, right? The magic happens, the peptide is loaded, and then these vesicles come back to the membrane to eventually present this to T cells. Okay, so this is the general process. So a great question, thanks for asking. And if there are any more questions, please send me an email. Thanks, bye.